In this video, we're going to learn about the process of transcription, which, as we learned in the previous video, is the process of reading a single-stranded DNA template and using this to produce an mRNA or messenger RNA transcript. Now, the first burden that needs to be overcome in terms of producing mRNA is that we need to identify where the genes are. Recall that a gene is any sequence of DNA that is ultimately used to produce mRNA, which then is ultimately used in the next stage, translation, to produce a protein by telling the ribosomes how to do that. The problem is, as we already established, not all sequences in the DNA are actually genes, which means that the enzyme that regulates transcription needs a way to recognize where a gene starts and where a gene ends. And the starting sequence to every gene is a specific DNA sequence that we call a promoter. So in colloquial English, the verb form of this word, to promote, means to advertise or to draw attention to something, and that's exactly what this regulatory sequence of DNA is designed to do. Its job is to draw attention to itself and attract the primary enzyme that is used in the production of mRNA, and indeed all RNA, that being RNA polymerase. So not only does RNA polymerase have the ability to recognize the specific promoter sequence using its active site, but hormones that are directly responsible for regulating the amount of protein that is available can either make the promoter easier to bind to RNA polymerase, thereby increasing the amount of a specific protein that is produced, or decrease the degree that the promoter is able to attract RNA polymerase, effectively shutting the entire process off. But I've moved ahead of myself. Now we need to talk about how RNA polymerase knows where the end of a gene is, and not surprisingly, there is a second regulatory sequence whose job it is to tell RNA polymerase that the gene ends at a certain point and to stop all activity with producing mRNA, and that, not surprisingly, is what we call the terminator or the termination sequence. So once RNA polymerase recognizes that there is a promoter region and that is the beginning of the gene, that is the signal for RNA polymerase to begin the 2 points of activity that it is able to do. So in the same way that DNA replication requires a single strand, meaning that the two DNA strands of the double helix need to be open, and again, this is what helicase does, RNA polymerase actually combines helicase's responsibility with its second duty, so we don't actually need two separate enzymes here, because RNA polymerase here, we can see, is breaking the hydrogen bonds within the DNA double helix in order to get access to the single-stranded DNA template. Now from there, once the DNA double helix is open, the primary function of RNA polymerase is to use the single-stranded DNA molecule that it is bound to here as a template in order to produce the new RNA strand. So its job, which works exactly the way that DNA polymerase does, is it reads the template strand and adds new nucleotides that are complementary to the DNA strand that it is using as a template, which is what we can see here. So here, as RNA is opening the DNA double helix here, or RNA polymerase rather, is opening the DNA double helix here, we can see that all of the nucleotides that are being added to the growing mRNA molecule are all complementary to the DNA bases that we see in the template. In other words, it adds nucleotides the same way that DNA polymerase does by using complementary base pairing. Now, there is, of course, one difference between the complementary base pairing of DNA and RNA, and that is that DNA contains thymine, but instead of adding thymine to an RNA molecule, uracil would be added instead. But otherwise, guanine and cytosine pair with each other like this. We can also see here that the new end of the mRNA molecule is the 3' end here, and the 5' end is actually the beginning of the mRNA molecule, which means that RNA polymerase also works using the same mechanism and therefore in the same direction as DNA polymerase does, in that it starts with the 5' end and moves in the 3' direction, because the actual reaction that it catalyzes is the joining of a new nucleotide to the OH group on carbon 3, the same way that we saw with DNA polymerase. Now, eventually, the RNA polymerase molecule, once it gets from the promoter all the way to the terminator sequence, eventually that terminator sequence is going to tell RNA polymerase to deactivate, meaning to stop its function, and when it deactivates, the RNA polymerase will actually fall off of the DNA strand, and if the RNA polymerase molecule is not here, there is nothing to hold the two strands of the DNA double helix together, and therefore the DNA double helix closes to the way that it was before transcription begins, and the mRNA molecule separates and ultimately leaves the nucleus and journeys towards the endoplasmic reticulum, where it will either be transported to cytoplasmic ribosomes or just transcribed by the ribosomes or translated by the ribosomes in the RER into protein, but that is the subject of a future video. Now, between that process, sometimes the mRNA molecule that comes out of this process will be done and perfectly ready to be used, and therefore transported directly out of the nucleus. But other times, depending on what the gene is, the mRNA transcript may undergo some sort of modification depending on what the specific protein is that is needed, and this type of modification involves either selective removal or selective modification in some way to the actual mRNA sequence, and this is a process that we call splicing, which is the subject of the next video that we will watch in this series.